Inflammasomes are intracellular multi-protein complexes comprised of three components. First is a unique sensor molecule like NLRP3. There are at least 6 different sensor molecules, each stimulated by different pathogens or internal stressors. Inflammasomes are named after the associated sensor molecule. This is an NLRP3 inflammasome. Second is adaptor protein ASC, associated with all 6 types of inflammasomes. Third is enzyme procaspase-1.
How are inflammasomes formed and activated to trigger inflammation?
In the presence of relevant pathogens or other inducers, the sensor molecule is stimulated, triggering a conformational change and oligomerization to form a wheel-shaped signaling hub. This hub recruits ASC, which also begins to oligomerize, and recruit procaspase-1. The inflammasome continues to recruit ASC and procaspase-1, forming a large filamentous signaling platform known as an ASC spec. This provides a scaffold for recruitment of more procaspase-1 molecules and triggers conversion of procaspase-1 to its active form, caspase-1, which is released from the spec. Caspase-1 cleaves cytokine pro-IL-1 beta to its active form, IL-1 beta, a powerful stimulator of inflammation. Inflammasomes not only initiate intracellular inflammation, they also promote extracellular inflammation by releasing ASC specs outside the cell. This perpetuates an ongoing damaging inflammatory response, contributing to a variety of inflammatory diseases. Targeting inflammasomes to control inflammation before it becomes pathogenic is a promising therapeutic approach for millions with inflammatory disorders.
IC100, a monoclonal antibody, is a promising approach to treating inflammatory disorders. IC100 uniquely binds to ASC proteins and inhibits ASC recruitment by activated NLRP3, thereby preventing initiation of inflammation. IC100 also binds to ASC in intracellular and extracellular ASC specs, preventing further recruitment of ASC and procaspase-1. In doing so, IC100 interferes with ASC spec function, blocking perpetuation of chronic damaging inflammation. Because ASC proteins are associated with at least 6 types of inflammasomes, IC100 has potential to treat numerous inflammatory disorders associated with activation of one or more types of inflammasome. In summary, IC100 is a novel ASC inhibitor that blocks inflammasome formation, preventing initiation of inflammation. It also interferes with ASC spec function, blocking perpetuation of inflammation. This controls chronic inflammation that contributes to disease.